Okay. All right. A warm welcome, everyone, to this new webinar of the Origins Center. I'm Bob Planquet. I will be chairing today's event. We're joined today by scientists through Zoom and also by some people from the general public through a live stream on YouTube. And I've seen there are already nearly a dozen people there. So warm welcome to you all. Great to see you all here. Just to explain a little bit, if you have never been to one of these webinars of ours, the Origins Center is a Dutch initiative which tries to bring together scientists from many different disciplines that are working on some of the most fundamental questions, the origin and evolution of life on Earth and in the universe. In this webinar series, we invite leading researchers from around the world, from physicists, astronomers, chemists, biologists, and scientists from all stripes. And today we have Dan Needleman as our speaker. But before I introduce him to you, let me remind you of the program of today. First, we'll have Dan's talk for about 45 minutes or so, and then there will be time to ask questions until roughly 5 o'clock. You can post your questions in the chat, both in Zoom and on YouTube. I'll try to keep tabs. You're, of course, also welcome to try to intervene a little bit. Everyone has lots of you have their cameras on and you can just speak. We're not with 150 people, so try to keep things informal. But of course, if you're on YouTube, if you're watching, you can also ask questions, but then you have to do it in the chat and I'll try to post the question for you. And at around 5 o'clock, we will close the session and the live stream on YouTube will be closed. We will stick around a bit afterwards. So if you have time for some social interaction, then do stick around. A couple of you have registered to continue discussions with Dan after 5 o'clock, and we'll see how we will do that, if we have to make a little breakout room to talk more privately, or whether we can just do it in this arena. We'll see how it goes. But for those of you, just stick around, of course. All right, so we're very happy to have Dan Needleman from Harvard with us today. Dan runs a great lab in which physics and cellular and molecular biology techniques are used to study self-organization in complex living systems. And his lab is a good example of the interdisciplinary nature of origins-related research, combining theory and experiments and involving people from the very diverse scientific backgrounds. And his talk is called Biophysics and Evolutionary Genetics of Cell Division. Please, Dan, the floor is yours.
Great, thank you so much. Yeah, it's a real pleasure to be here and to have the opportunity to give this talk, this presentation. And yeah, it'd be great if if people have questions, please don't hesitate to shout out or type in the chat or whatever. That'd be wonderful. All right, so I'm going to tell you guys about work that we've done combining biophysics and evolutionary genetics. And so this is this work is a collaboration of many people. And then the main hero of the story is Reza Fahardafar, who used to be in a postdoc in my group, who's now a research scientist at the Flatiron Institute. All right, so my group works on a number of different topics, and one of the main things we've done work on and are continuing to work on is cell division in the spindle. So the spindle is the self-organized structure that segregates chromosomes during cell division, and it's also involved in positioning the cleavage plane. So the position of the spindle determines the position of the cleavage plane in many contexts. And so a lot of what we've done in my lab and a lot of what we do in my lab is try to understand kind of mechanistic biophysics of how the spindle works, basically. But in addition to that, I'm very interested in the fact that spindles in different organisms are really quite different from each other. So the spindle, they can have very different sizes, very different shapes, very different dynamics. And it became very interesting in trying to understand this and trying to understand this in many different senses. So trying to understand what's the evolutionary basis of these differences. So what's the role of selection and genetic drift and mutations and other, evolutionary factors? What are the mechanistic basis of these differences, in terms of like genetic factors and biophysical differences? And also trying to use differences between spindles and different, individuals or organisms, try to leverage that difference to learn more about mechanistic aspects of spindles. And that last bit is what I'm going to be focusing most on today. Okay, so And I'm going to be talking about spindle elongation and positioning. And in particular, like a lot of what I'm going to discuss is going to be about the first mitotic division in C. elegans, which is, you can see a movie of right here. We can see the spindle, starts in one position, moves around, elongates, undergoes, interesting motions, and segregates chromosomes. And so, as I said, like you can talk about the elongation of the spindle, which is one thing which we'd really like to understand. And in particular, what determines spindle length. And what I mean by that is you can see that in this movie and as quantified in this graph here, the spindle starts at some initial length, it elongates at some rate and gets to some final length. And so what determines this initial length, what determines this elongation rate, and what determines its final length? This is, things we'd all like to, we'd like to know. And in this talk, again, I'll touch on some of these other ones, but I'll focus mostly on trying to understand what determines the final length of the spindle after the elongation ceases. So that's elongation, but also I'm going to talk a little bit about positioning. So if you look at the center of the spindle along this kind of what I'm calling the x-axis, a long axis of the embryo, you can see it undergoes this kind of complicated dynamics. And at the end of this, the final position of the spindle is asymmetric. So this first mitotic division in C. elegans is asymmetric. The division isn't in the middle of the cell, and the position of the spindle is not in the middle of the cell. And so we want to understand what determines these kind of complicated dynamics and what determines its final position. All right, so I'm going to back up again and talk about the first bit, which I mentioned, which is spindle size regulation and scaling. So many different classes of models have been proposed to try to explain the size of the spindle. So some people have suggested that spindle size is set by a timer. In other words, size is set by regulating, like when the growth of the spindle stops. So that'd be kind of a timer model. Other people have proposed kind of limiting component models, which suggests that, the spindle continues to grow until all of some like limiting molecule is used up, and that's what determines the final size of the spindle. Other people propose models in which the spindle kind of grows until it gets a fixed distance from the boundary, and that's what determines the size of the spindle. And I'm sure that you can think of many other additional possible models, and other people have proposed many, many other models. And so there's a whole range of possible models, kind of general classes of possible models of what determines spindle size. Okay, and the question is, how can we systematically test which of these possible models, including other ones we might not have thought of, are really important for determining are really like the main mechanism for determining the final spindle length in C. elegans. And what we decided to use is natural genetic variation in spindles to help answer this question. So try to test kind of all of these models simultaneously. All right, what do I mean by that? What I mean is that we decided to investigate spindle size regulation and scaling using recombinant inbred lines, which were lines created by Matt Rockman, who's now at NYU. And so what Matt did is that he created a bunch of lines generated by crossing two C. elegans wild isolates. So he took two isolates. One is a N2, which is what people typically use as a standard lab strain, and that was isolated in Bristol, I believe. And then he took another wild isolate of C. elegans, CB4856, which is from Hawaii, which at the time when he made these lines was the most genetically distinct, the most genetically divergent line of C. elegans, which people had collected, genetically divergent from the N2 strain. And then what he did is that he took these two and he crossed them together, and then he crossed their kids together. And then he crossed their kids together and their kids together and their kids together. He had a total of 10 generations of crossing. And then he took those lines and then he selfed them for 10 generations. And this ended up with 200 recombinant inbred lines, which are kind of homozygotic at basically all regions, which have different combinations of alleles from the original two lines. All right. And now it ends up, okay, so like Matt made these lines originally for his own region. It ends up that if you look at the spindles in the original two lines, N2 and CB4856, they actually look very similar to each other, which isn't a promising start for doing genetics. But nonetheless, when Reza looked at these lines, or looked at spindles in these recombinant inbred lines, he saw that, for example, some lines have very small spindles and some lines have very large spindles. In other words, the RILs have very different spindles in these recombinant inbred lines, even though the initial two lines, which kind of were used to generate all these recombinant inbred lines, the spindles are very similar. So that means that those two initial lines have similar looking spindles, but for different genetic reasons. All right. And then what we decided to do was basically quantify spindle size and dynamics in all these 200 recombinant inbred lines. All right, are people with me so far? All right, awesome. All right, so then what Reza did was he developed a way of doing high throughput imaging of the first mitotic division in C. elegans. And so here I'm showing multiple C. elegans embryos being imaged with DIC. So Reza did 3D imaging. This is just showing one Z slice of a 3D movie. And he imaged 50 embryos per line for each of these 200 recombinant inbred lines. So that's 10,000 embryos in total for high resolution time-lapse movies. That's a lot of imaging. And there's no way that Reza or anyone else could analyze these movies by hand. And therefore what Reza did is that he developed an automated image analysis platform to make sense of these movies. And using this, he was able to track, here you can see the spindle poles in the spindle itself automatically. And so for every embryo, he can track how the spindle elongates as a function of time. This is data from this one particular embryo. You can see that as it's elongating, as it's moving, it kind of reorients and it kind of rots. In other words, the spindle oscillates, so you can track those spindle oscillations automatically. And he can, therefore quantify spindle elongation, positioning, oscillations, centrosome size even, and many other factors, and get this kind of very quantitative data for each of these 10,000 embryos in each of these 200 lines. All right, so probably the most important take-home message of this talk is that he found that there's quantitative variation for all aspects of cell division and spindle length and dynamics across the recombinant inbred lines. That means that there's genetic variation for everything that he can measure. All right. And I just want to okay, so what does that mean? Oh, okay, well, first I'll give one example. So this is looking at the final spindle length for each of the 200 recombinant inbred lines. This is the average final spindle length from that line for each of the 200 recombinant inbred lines, just rank ordered by final spindle length. And you can see that some lines have very short final spindles and some lines have very long final spindles. Again, what that means is that, so what's different between the two lines? Well, the difference is that they have different combination of alleles from the original parent lines, and hence this variation is due to the combined effect of many small genetic differences between the lines. So again, that means that there's genetic differences, there's within species genetic differences for final spindle length, and for every other aspect of spindles that Reza can measure. So just like, like different people, there's genetic differences in, height and size of our noses and size of our ears and all sorts of factors. There's also genetic differences in all the kind of cell biological traits, at least that Reza measured, and presumably many, many, many more. So personally, I think that's a very interesting, important take-home message. All right. And the other thing is that we can now use these quantitative variations to test possible models of spindle size regulation and scaling. All right, what do I mean by that? Okay, so let's consider one particular possible model of spindle size regulation and scaling. All right, so let's consider a timer model. All right, so what do I mean by a timer model in this context? My definition of a timer model for final spindle length is to say that, okay, there's one set of genetic mechanisms which determine the initial length of the spindle, and there's other genetic mechanisms which determine how how long the spindle is going to elongate for, that's timing, and the speed it's going to elongate. And these are like all different genetic mechanisms. And so in other words, the final spindle length results from, genetic factors that determine initial length and genetic factors that determine the extent of elongation. That's my model for a timer. All right, we can say, is the data consistent with this model or not? How can we test that? Well, if this model was correct, this would predict that if you look across the lines, because the genetic factors by definition that determine in this model, by definition, the genetic factors that determine initial length are different from the genetic factors that determine the extent of spindle elongation. I can see from this proposed model that if this model is correct, lines which tend to have a longer initial length will tend to have a larger final length. That means that this model predicts that if we look across the lines, there should be a correlation between initial spindle length and final spindle length. Right? And then Reza looks across the lines and sees, in fact, these two are not correlated. That means just by looking at this, lack of correlation, we can essentially rule out all sorts of different particular timer models that fall under this definition, which is a wide range of possible specific models. So does this make sense to people, the logic of the argument? If not, please shout out. Okay. All right. So we can reject this model. All right, let's consider another possible model. All right, so another possible proposed is this limiting component type model. And again, in this context, the simplest version of limiting component model is to say there's one set of genetic mechanisms which determine the initial size of the spindle. And then, like I said, the spindle elongates until it uses up all of some limiting component. If we assume that another set of genetic mechanisms determines the concentration of that component in cells, and then a separate set of genetic mechanisms determines the size of the cell. Again, it's, I'm just considering this particular model right now. You could imagine others, but this is a model that I want to consider. If this was the case, then this says that the final spindle length is determined by, the concentration of the limiting component and the size of the final volume of the cell. And I'm postulating in my version of a limiting component model that the concentration of this limiting component and the volume of the cell are controlled by different genetic factors. If that's correct, then this model predicts there should be a correlation between cell volume and the final length of the spindle. All right, is that true? Yes, there's a very strong correlation between the volume of the cell and the final length of the spindle. All right, however, cell volume is also correlated with cell length. All right, so if all I know is that cell volume is correlated with final spindle length, like, is it really volume that's the important thing, or is it cell length that's the important thing, or is it both? How can we tell? Well, you can just do the following. You can say, let's look at the contribution of cell volume if you condition on cell length, right? So you're saying basically, if you look at cells which have the same length and different volumes, i.e. different widths, for example, is there a dependence with spindle length? And the answer is no. All right, so once you condition on cell length, cell volume is not correlated with final length. What that shows you is that the important thing isn't volume, it's cell length. All right, that means that we can rule out, again, the simplest version of a limiting component model. All right, so now we know cell length must be an important thing. Now let's consider another class of models, this kind of so-called boundary models. And again, in this context, my version of a boundary model is to say that, all right, one set of genetic mechanisms determines the initial length of the spindle, and then the spindle elongates until it reaches some fixed distance from the cell boundary. And there's a centrosome distance, which is the final distance of the centrosome from the cell boundary. And I'm imagining that there's some different set of genetic mechanisms that controls that final centrosome distance, and another set of genetic mechanisms that controls the length of the cell. Right. And if this version of a boundary model is correct, that predicts that the bigger the cell is, the bigger the final spindle length is. That was kind of implicitly true based on the data which I showed you before, and you can show that it's explicitly true. And this also predicts that in this class of boundary models, that the bigger the cell distance is, the smaller the final length of the spindle is, right? So for a given length of the cell in this model, if this distance is larger, then the spindle is going to be smaller, right, in this class of boundary models. So the boundary model predicts a negative correlation between final spindle length and the final distance of the centrosome to the boundary. Is that true? No. All right, so the final distance of the centrosome to the boundary is actually positively correlated with the final spindle length. All right, so that means that we can rule out the simplest version of the boundary models as well. Okay, so we can rule out a bunch of very generic classes of models, and we're not left with anything. So that's a little bit confusing. And so what we decided to do, like people often do in biology, if you're confused, and that is search for molecular factors that have an impact, and maybe that will give some insight on what's going on, right? So if we rule out models, don't know what to do, let's look for the genetic, see if we can see something about the genetic basis of spindle size regulation and scaling by looking for associations between genotype and phenotype. So we have measures of all these quantitative traits across these 200 recombinant inbred lines. Each of these 200 recombinant inbred lines is sequenced. Therefore, we know which kind of alleles come from the two parents. And now we can associate genotype and phenotype and see if we see anything. So for example, we can look at different regions along the chromosome and ask to what extent those regions are significantly associated with variation, in this case, in final spindle length. And here we see 1, 2, 3, 4 different regions, different chromosomal regions, which explains significant fractions of variance in final spindle length across the recombinant inbred lines, right? That means, presumably, there are some molecular, genetic factors in those regions which are contributing to final spindle length, right? That's interesting. And so I said before, cell length seems to be important. If we do a similar thing and look for genetic factors that influence cell length, we also see different regions that are, we see regions that are significantly associated with variation in cell length. And we see that there are common loci influencing both final spindle length and cell length, which is consistent with the idea that these loci are kind of like impacting cell length, and because they impact cell length, they indirectly affect the final spindle length. But now we can ask for something more complicated. We can say, okay, that's great, but I already know that cell length is important. Let's try to look for genetic factors that influence final spindle length independent of cell length. And so what we can do is that we can look at a kind of a new trait, which we can kind of construct, which is final spindle length conditioned on cell length. And we can look for genetic, we can look for regions across the chromosomes that are associated with that. And we see one very significant QTL, which people call these genetic regions QTL, or quantitative trait loci. And we see one region which is very, very highly correlated with variation in final spindle length that's independent of cell length. All right, and if we condition on this, so we're looking for now factors that influence final spindle length independent of cell length and QTL1, we see another QTL, QTL2. Right, and And again, if we condition on QTL2, we see no more QTLs. Very interestingly, these two QTLs seem to have opposite effects. So in other words, recombinant inbred lines with the N2 variant of QTL1 tend to have smaller spindles, and recombinant recombinant inbred lines with the N2 variant of QTL2 tend to have larger spindles. So this is kind of consistent with a picture where, again, you have these original two lines which have different genetic factors contributing to spindle length, but those genetic factors are somehow basically kind of canceling each other out to produce what appears to be similar looking spindles, again, for different genetic reasons. All right, so what are these factors? All right, so what is QTL1? So each one of these regions actually contains, are relatively large, and there's multiple genes under each QTL. But sitting right smack in the middle of this QTL is a gene called GPR1, which people who work on the spindle and C. elegans are very, very familiar with. Interestingly, there are two non-synonymous variants in GPR1 between these two strains. Consistent with what people have known for a long time, if you knock down GPR1 with RNAi, you reduce the final length of the spindle. All right, so this is consistent with GPR1 being QTL1. That's not a proof of it, but it's consistent with that. All right, what does GPR1 do? Based on a lot of work from a lot of people, GPR1 is known to localize or to interact with the cell cortex and to activate dyning, which is a molecular motor on the cell cortex. Okay, this is arguing that cortical forces, pooling forces, are likely going to be important for controlling final spindle length. All right. Now, all right. So that's suggesting that's what GPR1 that's what QTL1 is. What's QTL2? All right. Sitting right under QTL2 is another protein. Again, there are multiple genes under there, but one of the genes under there is one called PAR2, which again, people who work on C. elegans spindles are familiar with. And interestingly, if you look at these two lines, there's a very large, very significant difference in expression in PAR2 between the two parent lines. And so perhaps this QTL is controlling the expression of PAR2, or this is consistent with the idea that this QTL is controlling the expression of PAR2. If you experimentally knock down PAR2, you see that influences final spindle length. All right. So what does PAR2 do? PAR2 actually is involved in controlling the localization of GPR1. So actually, if you, based on a lot of work from a lot of people, if you look at the first mitotic division of C. elegans, there are a variety of different PAR proteins which are involved in setting up two cortical domains in the embryo, and PAR2 is one of the ones which are kind of responsible for this. And like I said, it localizes GPR1, so this is consistent with the PAR domains and cortical forces controlling final spindle length. All right. So based on analysis of the RILs, we could rule out a wide variety of models of what controls final spindle length. And we're left with that, well, okay, cell length is clearly a very important thing. GPR1 is clearly an important thing, or the sequence of GPR1 is a very important thing, and the expression level of PAR2 is an important thing. Right, so this suggests that the final spindle length is perhaps set by a cell length dependent balance of forces. All right, so what are these forces? What forces set final spindle length? And these are the forces, and what are the forces acting after elongation has ceased, right? So, you could talk about the forces that cause elongation, that's very interesting, but here we're talking about what are the balance of forces after elongation has ceases that determine the final spindle length. That's what we want to gain more insight into. All right, so to do this, two graduate students in lab, Haiyan and Chi Hong, built a laser ablation system, which I humbly believe is the best laser ablation system in the entire world. It is incredibly impressive. It involves a lot of programming. It uses a reduced repetition rate femtosecond laser to do highly precise cutting in nearly arbitrary three-dimensional patterns. So for example, here's a picture of a C. elegans embryo or movie, and whenever you want, you can cut, and see what happens. All right. So now Reza used this laser ablation system to do different types of cuts of microtubules after the spindle had ceased elongating. So when the spindle is at its final length, in order to learn about the forces acting on the spindle. So for example, right, what are the forces? The idea is to use laser ablation to dissect the cortical forces which determine the final spindle length. So for example, one hypothesis, one model you could imagine, is that there's a balance between pushing and pulling forces that sets the distance between it sets the final distance between the centrosome and the cell boundary, right? So in other words, maybe the centrosome elongates until, so you have all these microtubules reaching out, hitting the cell boundary. Maybe some of those push, some of those pull, and it's those those pushing and pulling forces come in balance, and that determines the final spindle length, the final position of the centrosome. All right. Now, that would predict that if Reza laser ablates kind of a cup, right, and cuts all the microtubules besides those in this cone, then the centrosome should remain balanced, because in this model, it's only the microtubules that remain over here which are determining the final position. All right, so now he's going to cut a cone, and we'll see what happens. All right, so the spindle elongates. After it reaches its final length, bam, he cuts, and you see the centrosome flies towards the cell boundary, right, after the cut, after the spindle ceased elongating. So what that means is that in this final position, there's not a balance of pushing and pulling forces from microtubules pointing in that direction. Rather, there's net pulling forces from microtubules in that direction. All right. So if there's net pulling forces from microtubules in that direction, at the end of elongation, the centrosome isn't moving. That means there must be zero net force acting on it. That means those net pulling forces from those microtubules must be balanced by some other forces. What are those other forces? Where do they come from? A natural guess is maybe it's the elasticity of the spindle itself, right? So maybe the spindle itself acts as a spring. If that were true, then cutting the spindle itself should break that spring, and then the centrosome should again fly towards the cortex, just as in the movie we just saw. All right, so Reza did that experiment as well. So he watches the spindle elongate, waits till it gets to its final length, and then cuts spindle microtubules, basically cuts the spindle apart, and the centrosome essentially doesn't move anymore. All right, so what that means is that he's cut the spindle in half, but the centrosome doesn't move. That means that the spindle elasticity does not significantly contribute to the final spindle length. So it is in fact not a balance of spindle elasticity and pulling forces. All right, what could be, if that's not it, what else might be going on? Maybe what's happening is that you have microtubules in different directions, which are all exerting pulling forces, right? So you have some microtubules pulling this way, some microtubules pulling this way, because of their contact with the cortex, and maybe all those pulling forces become in balance. And it's the balance of those pulling forces which determine the final position of the centrosome after the spindle elongates. All right, if that were true, if there really were pulling forces in all directions, then if Reza does a cup cut in this direction, then the centrosome should fly toward that surface because there should be pulling forces in that direction. Is that happening? So after the spindle elongates, he cuts, and the centrosome does fly. So in fact, there are pulling forces in all directions, which is consistent with this with a model where, the final position of the centrosome is set by a balance of pulling forces from different directions. All right. Okay, so now if that's really what's going on, then that predicts that the central spindle should actually have no impact, the presence of the central spindle, on the position, on the final position of the centrosomes and on the final spindle length. And so one can test that using an RNAi, RNAi the protein PRC1. Oh, oops, somehow, shoot. All right, these movies, okay, here we go. All right, so if you RNAi PCR1, then what happens is that the spindle breaks in two. And you don't have a central spindle, yet the final length of the spindle, I mean, the elongation dynamics are affected, but the final length of the spindle that the spindle goes to is the same. So in other words, even if you don't have a central spindle, you still get the same end position of the centrosomes. In other words, the spindle is not important for determining spindle length, or at least final spindle length, right? This is consistent with a picture, which I was just saying, where it's really a balance of pulling forces acting on the centrosomes after the end of elongation. Okay, but, all right, so is that really possible? Can a balance between lateral and longitudinal pulling forces actually stably position centrosomes? So we work with Mike Shelley, who's at Flatiron NYU, to come up with a biophysical model of how cortical pulling forces can stably position centrosomes. And I'll go through this kind of quickly in the interest of time, but the gist of it is that if you have a model where you have kind of cortical force generators, the microtubules can grow out and hit those cortical force generators, and you assume that, so you have microtubules can grow with some velocity vg, they can catastrophe with some rate lambda. If they bind to a force generator, they experience a constant force F naught going toward that force generator, and those force generators are some size r. And if they're bound, they can unbind from the force generator, they can detach at some rate k. And you assume, and this is totally critical, that only one microtubule can bind to a force generator at a time. If you assume that, then you can write down a model of the force that a force generator exerts on the astral microtubules, which is based on the probability that an astral microtubule is attached to this. And so in other words, any given, I'll just say this again, that any given microtubule that is attached to the force generator generates the same force, F naught, but depending on the distance that the centrosome is away from the force generator, the probability that it is attached by a microtubule will change. And you can solve for this probability, and then if you take this model and you kind of imagine you have a bunch of centrosomes all over, you have a bunch of force generators all over the cortex, you can write down what's the total motion as a balance of forces. And one can kind of computationally solve this in 3D. And in this model, which only has pulling forces, no pushing forces, no bending forces, nothing else, only pulling forces, you get that centrosomes can stably position. And I'll just note that some people have proposed a model where that pulling forces could stably position, and that's based on having a limited number of force generators. And I'll just note that's not what's happening in this model. And in fact, this model, you can stably position centrosomes even if you have more force generators than microtubules. This is kind of a side note. And what's going on in this model is that basically what happens is that if the centrosome displaces, imagine like here's a cross section of the embryo, the centrosome is up a little higher, the force generators on top will actually generate more force upwards because they're more likely to be bound, but the projection of that force in the upward direction is smaller because this force is at a more oblique angle. And because of these two effects, because of this geometry, this oblique angle, and because only a force generator can only bind one microtubule at a time, just having cortical pulling forces can be stabilizing. one interesting prediction of this model is because in this model, only one force generator can bind a microtubule at a time. That suggests that if you have two centrosomes, like you really do, they can kind of compete for force generators. And what that means is that that argues that removing one centrosome will impact the contribution of force generators have on the other one. And so if you have two centrosomes and you remove one, the other centrosome should migrate to the center of the cell. That's kind of a prediction of this model that's very, very non-trivial. And we can test that experimentally by using laser ablation to blow up one centrosome. And then you see that the other one moves over. All right, so that's a strong argument. Now, in this model, I'm not going to wrap up very quickly. In this model, so sorry, and we know that in reality, there's a lot of work from a lot of people to say that you actually have, based on PAR2, like I was saying before, in other PAR proteins, you have an asymmetric number of force generators on the two sides of the centrosome. If you put that in by hand, what you get, or if you put that in this model, what you get is that the spindle elongates asymmetrically, which in fact it does experimentally. And if you use, with parameters extracted from the literature, the theory can explain the full dynamics of spindle elongation. Here we have this theory compared to experiments. And in fact, the complicated positioning dynamics. Again, this is just using this very simple model I just said before. Furthermore, this model predicts that the correlation between final spindle lengths and cell size that is consistent with what we observe across the recombinant inbred lines. And not only does it predict the correlation between final spindle length and cell size, but also it accurately predicts the correlation between centrosome distance and cell size. It predicts that positive scaling I mentioned previously, how the spindle center moves when you change cell size, and how the elongation rate varies when you change cell size, all from this very, very simple model. And so this model does explain scaling of the spindle across the recombinant inbred lines. Furthermore, in a previous study, we collaborated with Marie Delattre and characterized nearly 40 other nematode species with known phylogeny that span over 120 million years of evolution. And we studied multiple wild isolates for many of these different species. And what we found in that earlier study is that many aspects of the spindle, if you compare across species, also correlate with cell lengths. And in fact, if we look at the correlations which we observed across species, those are consistent with what we see with the RILs and with theory. So this theory can explain the scaling of cell length with the scaling of spindle lengths with cell length. Sorry, the scaling of final spindle size with cell length, the scaling of centrosome distance with cell length, the scaling of the spindle's positioning with cell length, and the scaling of elongation with cell lengths across these roughly 40 different nematode species. All right, so then in conclusion, I'll just say that there's extensive within species genetic variation for cell biological traits for basically every aspect of the spindle that we looked at. A model with only cortical pulling forces in which each force generator can only pull on one microtubule at a time can quantitatively explain the fact that you get stable centrosomes, that you get stable centrosome positioning and spindle positioning, as well as the time course of spindle elongation and positioning, as well as the scaling of the spindle across the recombinant inbred lines in nematode species. And I'll note that in previous work that we did, we found very strong evidence that cell size, and in previous work where we looked at within species variation of spindles and between species variation of spindles in nematodes, we found very strong evidence that the size of the cell is subject to stabilizing selection, and that stabilizing selection on cell size is sufficient to explain within species and between species variation in spindle traits and C. elegans and other nematodes. And that stabilized selection could explain that because those traits were correlated with those spindle traits were correlated with cell size. And now here, what I've told you, we now have a kind of a mechanistic understanding of those correlations. So I would argue that this is essentially a mechanistic explanation for the differences in spindle traits across nematode species. All right, thank you so much. Oh yeah, this work was, like I said, Reza's a hero. There are many other people, and there's funding.
Thank you very much, Dan, for this wonderful talk. I'm sure I speak for all of us with this really impressive talk and series of, especially these experiments with this laser ablation. Looks really amazing. So are there any questions? I see Gautam raised his hand. Go ahead.
Thanks. Yeah, it was an amazing talk. So I was just wondering whether you had thought about syncytial systems where there are multiple spindles sharing the same cytoplasm. So your model would, of course, cortical forces are obviously important for positioning them, but now you would argue that even in such a system, it's cortical forces that would then determine the length of the spindle as well. So I was just wondering whether that was something that you'd looked into, or.
Well, yeah, and so one thing which I definitely think is true is that this explanation is not universal. So I think this is an explanation for variation of the first mitotic division in nematode species. It's certainly, I think, basically, I think it's very unlikely to apply, for example, in those type of systems. You presumably have other factors.
But why do you think that's so? I mean, that's certainly possible. I mean, evolution is pretty good at confounding us that way. But I guess, is there a specific reason you believe this is something that has evolved in the system?
Well, okay, so I'll answer that in two ways just to make sure I'm answering it properly. One statement is to say that I think there's unambiguous evidence that this can't be the explanation in other systems, because in other systems, like you were mentioning, you also can have in vitro systems where you can get a spindle to elongate and get a final spindle length, which is clearly not dependent on cortical pulling factor forces. So this can't be kind of a universal explanation. I think that's totally true. Now, a side note of what I said, so I went through this very quickly in the end, but what the conclusion of our kind of more evolutionary work was that what the data is consistent with, and I think this is I would argue we have a strong evidence that this is the case, is that really the main thing that's subject to stabilizing selection in this case, that's subject to selection, is just the size of the embryo. Or the size of the embryo, which it's consistent with a lot of other work that people have done, arguing that because the size in C. elegans, the size of the one cell stage is the size of the embryo, is the size of the organism at birth, and that is, size of organism at birth is definitely subject to stabilizing selection in many species, right? And therefore, I think that none of the spindle factors we are looking at are actually directly acted on by selection. I think it's just they kind of go along for the ride, like based on, like things acting on other things. So I think that the reason, so I don't think it's that this is somehow like optimized in this particular case. And it's going to, so like my picture then, broadly speaking, is I suspect that for the most part, you kind of have organismal kind of or life history traits being acted on directly by selection, and then that's kind of dragging along other factors just to like do as they do, even though they're not really being directly acted on.
Yeah, thanks.
Any other questions? I actually had a question on the very beginning. You talked about these, like the theories you debunked. There was, for instance, the one on the timer mechanism. So in that kind of pictorial depiction on what influences what, does that argument kind of assumes that the timer mechanism itself, the genetic mechanism that involves the timing, so how long these spindles grow, that is actually not subject to any variation and therefore constant between these different cell lines, or not?
No. So what the picture is, exactly. So the picture is that it's not subject to variation, but that it's different genetic factors which determine that, and that there's one set of genetic factors that determine that, and different set of genetic factors determine the initial spindle length. and that those two genetic factors are uncorrelated across the recombinant inbred line. That's what the assumption is. And so there still could be variation, and there kind of must be variation. If that model were right, there would be genetic variation for that factor, for the timing. But in that model, we're just proposing that it's uncorrelated from those that determine the initial length of the spindle. And so I think that's really, in my mind, I think those type of arguments I think are really interesting and really powerful. And one could always say, well, you know, like maybe your favorite timing model isn't of that nature, and that's fine. But like, you know, that's one class of timing model, the one I just proposed, you can rule out. One could think of other types of models and then ask, can you rule them out or not? And so then that's the way that I kind of look at it. Yeah.
It was all pretty convincing. Certainly the last model that seems to explain most of the dynamics actually involved. What's the source of the oscillations that of this positioning? Why does that? Do you have any idea?
Yeah, and so I mean, like a number of people have studied that, and people have proposed different models. We have unpublished work, which we'll hopefully submit in the near future, which argues that basically the same model can also explain that. With a slight modification, like the version of the model, which I again went over very quickly here, that I used to kind of plot that we used to plot the results here, was taken in a limit where the movement of the centrosomes are slow compared to the growing and shrinking of microtubules and the attachment and detachment of microtubules from force generators. And if you make that assumption, you can calculate like the stuff which I showed you. If you don't make that assumption, you can solve for everything numerically, and you can even do some stuff analytically, and you can show that the same model can give you oscillations. And the gist of it is, what can happen in this model, in that type of model, is that imagine, okay, on the one hand, these pulling forces want to stably position you, but then if you start moving up too quickly, let's pretend spontaneously, you kind of just kick the centrosome, it starts going up very quickly. What can happen is that it can move up so quickly that microtubules that grow down don't grow down fast enough to actually contact the lower surface, and therefore they don't have. Therefore, they basically don't have or have less of a pulling force on them below, and therefore that makes them want to move up. But then, due to this geometric effect, as they move up, the net force in the upward direction gets lower and lower, and therefore they slow down. At which point, microtubules in the lower surface can attach. And then you can start moving down very quickly, so quickly that you detach from the top, and that can give you these oscillations. And so like other people have proposed other models, but I think we have strong evidence that that's like the best model, but that's unpublished stuff.
So it's essentially a kind of delayed feedback, delayed positive feedback that trying to close a tap, but not getting a very quick reaction when you close it and open it again and close it again.
Yes, that's right. That's right. And that's related to ideas that Francois Needleman has proposed, also based on this kind of attachment to detachment. But this version of the model has aspects are different from the one that he proposed, but it's kind of related to some ideas that he has proposed as well.
Cool. Thanks. Any other questions? Also on YouTube, I just have a look again. So I did find out how to do this chat business globally, but I only found it afterwards. I know it for next time. So thanks, Dan, for this wonderful talk. And we're going to be sticking around here for just a minute, but for now, let me thank you all for being here. And this is the last webinar for this academic year, I believe, and we will continue in the next academic year. So I hope we will see many of you there again. And please stay tuned on the website of the Origin Center, and subscribe to our mailing list to be completely up to date of all the events that we try to organize. All right, bye-bye. Okay.